Hello friends, this video on Biomolecules Part 25 is brought to you by examfear.com. No more fear from exam. So let us now talk about the fatty acids in little more detail. There are two types of fatty acids. One is saturated fatty acid and the other one is an unsaturated fatty acid. Saturated fatty acid are those fatty acids which do not have a double bond. So the example which I mentioned before, if you consider this example, so this is a saturated fatty acid. So in this what happens, there is no double bond. Don't consider this double bond because this is for the acidic group, that is the carboxyl group. Other than this, there is no double bond. So this is a saturated fatty acid. So it is full of hydrogens everywhere. Whereas an unsaturated fatty acid will have a double bond. It can have one double bond, it can have two double bond, it can even have multiple double bonds. So let us take an example of unsaturated fatty acid. It will be somewhat like this. So this is unsaturated fatty acid. Why? Because you have a double bond here. The moment you have double bond here, the number of hydrogens will reduce because carbon at the max can have four bondings. So two here, one here, one here. So you cannot have any more hydrogen. So in this case, it was all full of hydrogen, but here it is not all full of hydrogen. Hydrogen is less, but double bonds are there. Now those unsaturated fatty acids where multiple double bonds are present, they are called polyunsaturated fatty acids. They are called polyunsaturated fatty acids. What are polyunsaturated fatty acids? They have multiple double bonds. So now we can say that these fatty acids are the monomers from which polymers can form, the way it was happening for all other biomolecules as well. By dehydration synthesis, polymers can form from monomers. So this carboxyl end is the acidic end, which is hydrophilic in nature, whereas the other end forms the hydrophobic end. These fatty acids can combine with other molecules to form substances, the way we saw in case of the complex lipids, the compound lipids rather, the phospholipids, the lipoproteins, the lipopolysaccharides. They are all formed by combination of these fatty acids with other molecules like proteins or polysaccharides or phosphates. So that is how they are formed. Now let us talk about each of the types of lipids in detail one by one. So we'll start our discussion with triglycerides. So we spoke about fatty acids, right? Now let us talk about triglycerides. So what are triglycerides? These are basically storage lipids. So storage is the main function of triglycerides. They are esters of fatty acids. Now what are esters? Let us see. Ester has come from the kind of linkage the fatty acids have amongst themselves to form triglycerides. So let us look at the structure of triglycerides. Now triglyceride consists of a glycerol, which is somewhat like this. What is a glycerol? It has alcohols. So it consists of one glycerol and three fatty acids. So one glycerol and three fatty acids together by dehydration synthesis will form a triglyceride. And the reverse process will take place by hydrolysis. So that is how the formation of triglyceride will take place. Now how exactly it happens? Let us see. Let us draw the structure for glycerol. So this is the structure for glycerol. This glycerol will combine with fatty acids. So this is how the fatty acids look like. Now since it will be combining with some three fatty acids, so the structure is not going to be very simple. It is going to be a complex structure. So these two are going to combine and one H2O will be removed and the structure which you get is the structure of triglyceride. So instead of drawing this, I'll show it to you on the next slide. This is how a triglyceride looks like. So if you see, you have R1, R2 and R3, and what are they? They are the fatty acids. So triglycerides, tri means 3. So it has 3 fatty acids and 1 glycerol. So by the process of dehydration synthesis, a bond is formed everywhere by removal of water. So this is the structure of a triglyceride. So let us now look at some of the functions of triglycerides. As I mentioned before, they act as good storage of energy due to their high concentration of carbon-hydrogen bonds. Since there are too many carbon-hydrogen bonds present, they help in storing energy. They act as insulators. Fat acts as an insulator. Why? Because they conserve body heat in animals. That is why you would have actually seen that animals which have a lot of fur, they can survive in extremely cold climate, whereas other animals can't. For example, for human beings, we cannot survive in climates where there is snowfall without sweaters. And what are sweaters? They are all made up of furs, so which give us warmth. So fats or triglycerides, whichever is energy storage, they also provide a lot of warmth, conserves body heat. They also act as shock absorber, that is fat present below the skin, they act as shock absorber. Shock absorber in the sense, have you ever observed that if you are carrying something very delicate, say something made up of glass, whenever you put it in your suitcase before you travel anywhere, what do you try to do? You try to wrap it in some cloth or some towel or some cotton so that it gets a cushion-like feel everywhere, so that even if somebody just throws your suitcase here and there, that material, the delicate material, is protected, because the cotton which you are surrounding over it will act as a shock absorber. So fat also play a similar role. It acts as a shock absorber and thus protects the underlying organs. They are solvents for vitamin A, D and K. Some of the sources of food which are rich in triglycerides or rich in fat are groundnut, ghee, butter, mustard oil. These are some of the sources which are high in lipids. So let us quickly have a look at some other lipids like phospholipids or steroids. So phospholipids, as I mentioned before, they are made up of 1 glycerol plus 2 fatty acids. Now in triglycerides, it was 3 fatty acids, but here there are 2 fatty acids. Since phospho is there, so there will be a phosphate group. So this is going to be the composition of phospholipids, a glycerol, 2 fatty acids and 1 phosphate group. Now here also the fatty acid tails are going to be hydrophobic in nature, whereas the phosphate group end is going to be hydrophilic in nature. So that will be the composition of phospholipids. Talking about sterols, how do you identify sterols? Wherever you have 4 carbon rings, you just recognize them as sterols. So presence of 4 carbon rings indicate a sterol structure. So let us look at some of the examples of lipids. So here you can see the presence of a carboxyl group which makes it a fatty acid, and this is palmitic acid. Glycerol, this is how it looks like. Triglyceride will have 1 glycerol and 3 fatty acids. That is a triglyceride. Cholesterol is nothing but a sterol because you can see 4 carbon rings here. Phospholipid, as I said, it will have 2 fatty acids, 1 glycerol which you can see here, and 1 phosphate group. So this is a phospholipid, that is lecithin in this case. So these are some of the examples of lipids. So with this we have reached an end towards our discussion on lipids. Now what is our conclusion from this entire lesson which we studied about all the biomolecules? We talked about carbohydrates, proteins, lipids and nucleic acids. So what did we understand? What is the we understood this, that okay, we understood the structure and functions of each of the biomolecules, which also saw that biomolecules play a very crucial role in the process of metabolism. So when we say that an organism is living, so what are we trying to say? What do we mean by the living state of an organism? So now all living organisms exist in a steady state characterized by concentrations of biomolecules. So the concentrations of biomolecules play a very crucial role. That is, as I mentioned you before also, for example, if you talk about lipids, if there is excess of lipids in your body, so you are not going to have a very healthy life. So there will be some or the other problems happening inside your body system. So the concentration of each of the body molecules, whether it is protein, whether it is lipid, whether it is carbohydrate, or it is nucleic acid, everything has to be present in the desired amount, in the correct proportion. Only then the organism will be living in a steady state, in a stable state. So when you talk about a human being, there are so many different processes which are happening inside our body. For example, blood circulation, or the process of respiration, or the process of digestion. But there has to be symmetry. Or now suppose if the process of digestion is happening, but if you do not have the enzymes, the digestive enzymes in the right amount, do you think that the process of digestion can happen in the right way? It can't. So when the process of digestion doesn't happen in the right way, all the systems of the body will get disbalanced, because when digestion is not happening, your body is not getting energy. So your cells will not get energy. Cells will not be able to perform their job of cellular respiration. So everything will get impacted. And when your cells are not functioning well, blood circulation is also not possible. So everything are in coordination with each other. So everything has to take place in the right way. So the concentration of biomolecules in our body is also very, very important. So we can say that living state is a non-equilibrium steady state to be able to perform work. So please remember that something which is living is in non-equilibrium. Why it is a non-equilibrium? Because since it is a non-equilibrium, that is why it is doing some work. Whenever it, for example, let us suppose you have a hot cup of coffee. So when there is a difference in temperature between the cup of coffee and the surroundings, there will be flow of some heat. So whenever both the temperatures will become equal, what will happen? There will be no flow of heat. So similarly, if the entire body is in equilibrium, there will be no work. Nothing will work inside your body. But there are so many things happening inside your body, that is because the entire state overall is in a non-equilibrium state. So that the entire body, all the biomolecules inside your body, they are able to perform some work and keep you living. So that is how a living state is defined. So with this, we have reached towards the end of this lesson, and I hope that this discussion on biomolecules would have helped you. And well, in this lesson, we don't have much questions to be included, because most of the questions have already been covered in whatever I have covered so far. So see you all in the next lesson. Thank you. Please visit www.examfear.com to watch more videos, attempt free online test, get free study material, find tutors and mentors. Thank you once again.